Hello, my name is Chris Campbell. I am presenting our work on structural connectome atlas construction in the space of Riemannian metrics. This is a joint collaboration across multiple institutions. Martin Bauer provided the mathematical foundation. Tom Fletcher and Sarang Joshi applied it to the structural connectome and tractography. Jose and Harsham Dai and I implemented the framework. There is a rich body of work that studies structural connectivity of the brain by analyzing tractography derived from diffusion imaging, including the work by Fan Zhang and Lauren O'Donnell et al., who produced the tractography shown here. However, a fundamental challenge remains open. How do we statistically analyze a population of these connectomes? I will present a novel framework to address this question. Our goal is to construct a structural connectome atlas from tractography data, where a structural connectome is a model of the white matter pathways between brain regions used to study the structural network of the brain. We construct this atlas so that we may statistically quantify the geometric variability of structural connectivity across a population. Methods exist to build white matter atlases. One method registers the diffusion-weighted image to an anatomical template by computing a Euclidean average of diffusion tensors at each voxel. However, this method does not take directionality information into consideration. Another method would register Q-space diffusion images to the anatomical template by averaging the spin distribution functions at each voxel, which does consider directionality information. However, there's no consistency of long-range white matter connections considered in this method. A third method would register the tractography itself directly, then cluster it into fiber bundles. This does consider the long-range connections. It's computationally expensive and can be manually intensive as well. In this talk, I will explain the following contributions in detail. Here we represent tractography fibers as geodesics of a metric, that is, as a point on the infinite dimensional manifold of Riemannian metrics. We use the diffeomorphism invariant Eben metric to compute distances of geodesics between connections. We provide a framework for diffeomorphic metric registration of connectomes and a method to then estimate an atlas of connectomes. So first, we need to represent the structural connectomes as Riemannian metric. Our goal is to find a metric whose geodesics match the tractography as defined by a vector field V. Note that this can be any vector field V that we're matching, not just one derived from, say, diffusion tensor imaging or other methods. So metrics that have been used before include the inverse diffusion tensor metric. The geodesics of this metric capture the essence of the tractography, but does tend to deviate from that tractography in high curvature areas. So instead, we want to build on the work from Howe et al. To estimate a locally adaptive metric, G alpha, that has a local conformal factor to the diffusion tensor metric, inverse diffusion tensor metric. This metric is chosen so that geodesics of the metric match the tractography. We do this by minimizing the functional shown. The solution equation that minimizes this functional is the Poisson equation. We solve that Poisson equation for the conformal factor alpha to get our locally adaptive metric. Here are some examples of our tractography-based metric estimation process. The tractography we are matching is shown in black on each image. Note that the blue geodesics of the inverse tensor do not match that tractography, while the orange geodesics do match it. The left metric is from synthetic data, while the center metric is an axial slice of data from a subject from the Human Connectome Project dataset. The right image is a close-up view of the GNU of the corpus callosum. So let's talk a little bit more about the manifold of metrics. The metric G belongs to the space of Riemannian metrics. Diffeomorphisms act on the space of metrics by pullback as shown. Notice here, T of phi is the push forward of phi, that is the differential d phi. Note that the geometries of G and phi star G are related via phi. In particular, geodesics with respect to G are mapped via phi to geodesics with respect to phi star G. We will equip this manifold with the Eben metric to get an infinite dimensional Riemannian manifold of Riemannian metrics. Let's talk a little bit more about the Eben metric, which is a metric on metrics. What is the Eben metric? Why did we choose it? Well, the Eben metric is the integral of pointwise metrics on the space of symmetric positive definite matrices. Note that this metric includes the volume density of the foot point G, which distinguishes this metric from the affine invariant metric others have studied. This dependence on the foot point gives us a reparameterization invariance under the action of the diffeomorphism group. The other key reason we choose the Eben metric is that it has explicit pointwise formulas for geodesics and distances. As you will see, this will allow for efficient calculations. Here is some mathematical detail on these explicit formulas. I won't go into it here, but I present it for those of you who are interested. Notice that the distance formula is an integral of pointwise distances on the space of symmetric positive definite matrices. Now, because it is the integral of these pointwise distances, we can visualize the distance between two connectome metrics. So I plot here the log of the distance for visualization purposes between the connectomes for two subjects from the Human Connectome Project dataset. The top row is, again, an axial slice for each subject, and the bottom row is a zoomed-in view of the generative corpus callosum. We can now formulate diffeomorphic metric registration using this mathematical foundation. First, we use the Eben metric geodesic distance to induce a right invariant distance on the space of diffeomorphisms. We fix a metric G, in this case, the Euclidean metric, in order to get a distance for diffeomorphism phi from the identity. We then construct an energy functional with two terms. The first is the regularization term on phi, which prefers diffeomorphisms that are close to the identity. The second term is the matching term, which measures how close the metric G1 deformed by phi gets to the metric G0. Note that we could easily extend this functional to include terms for point, curve, surface, or image matching. I will get into the algorithm details after I discuss atlas building. To build a connectome atlas for n subjects, we perform the following joint minimization. We find the metric G hat and the diffeomorphisms phi I that minimize the sum of distances from each deformed metric phi I star Gi to the atlas metric G hat. We do this using an alternating algorithm that was implemented in PyTorch. At each step, we alternate updating our estimate of the Frechet mean and updating our estimates of the phi Is. I'll go into detail on how we estimate the Frechet mean on the next slide. To update the diffeomorphisms, we register each connectome to the current estimate of the mean by running metric registration. The metric matching is a gradient flow optimization where we compute the push forward phi star G1, calculate the Eben energy between G0 and phi star G1, and then use PyTorch's automatic differentiation to help compute the gradient used to update the estimate for the diffeomorphism phi. Now we only do 2 iterations of metric matching for each step so that we don't overfit to early estimates of the atlas. To compute the Frechet mean, we use a recursive algorithm. Now recall that the Frechet mean is the metric that minimizes the sum of the distances from each of the sample metrics to that metric. This recursive algorithm works by starting with a first estimate of the mean as the first metric G1. Then we compute a geodesic between that estimate for the mean, and the next sample metric, G2. We step halfway along that geodesic, and that is our new estimate for the mean. We then compute a geodesic from that estimate of the mean to the metric G3 and step a third of the way along that geodesic. We repeat by computing a geodesic to the metric G4 and stepping a fourth of the way along that geodesic, and on to G5, we compute that geodesic and step a fifth of the way along. Note that this algorithm then converges to the estimate for the Fichet mean. The benefit of this recursive algorithm is that it requires only n geodesic calculations in total. We're using an efficient explicit geodesic computation and reducing the number of those computations. So this leads to a really efficient estimate for the Fichet mean. We performed some proof of concept experiments with synthetic and real data. For synthetic data, we generated vector fields whose integral curves come from a family of parameterized cubic functions. We performed 400 iterations of our atlas building algorithm with lambda equals 100 and a learning rate epsilon of 5. This learning rate is for the gradient flow, how much the gradient contributes at each step of the gradient flow. We found that this algorithm behaves particularly well when 1 over the learning rate is approximately equal to the Eben energy. Here's an example atlas of these synthetic tractograms. On the left, we have 4 sample metrics. We compute an atlas from the 4 metrics. Over here on the top, we plot that atlas and we pull with the tractograms from the individual subjects directly overlaid without deformation. We do this because the way that the data has been generated, we expect that the atlas geodesic will lie in the center of the 4 sample metrics. And we see that it does. This orange curve here is a geodesic for the atlas connectome. In the bottom plot, we see the same atlas again, but this time we have deformed the tractography from the individual metrics into atlas space. And we see how well those deformed tractograms line up with the geodesic in the atlas of the atlas. For real data, we used some subjects from the Human Connectome project. We estimate diffusion tensors for b value of 1,000. We performed 5,000 iterations of our atlas building algorithm using lambda of 100 and a learning rate of 1. We have found that lambda here helps balance the magnitude of diffeomorphisms from each connectome metrics to the atlas so that we don't get an imbalance where one connectome is being deformed a lot and another connectome is only being deformed a little bit. This way, all of the connectomes are being deformed roughly equivalent amount. Here is an example structural connectome atlas using 2 subjects from the Human Connectome project. On the left, you will see the diffeomorphisms from each subject to the atlas. In the center, you will see, again, an axial slice of the connectome metric for each of the subjects. And on the right, you see the atlas connectome metric. The top row is that axial slice. The bottom row is, again, the zoomed in version of the generated the corpus callosum. We take tractography from each of the sample metrics, and we map that into atlas space. The orange curve here is a geodesic in atlas space, and we can then look at how well those match with the subject tractography. So for future work, we want to really now get into the statistical analysis. Now that we can compute atlases, let's look at median or principal geodesic analysis or regression analysis for a population of connectomes. We also want to evaluate the robustness of connectome atlases to number of subjects and various subjects. How consistent are those atlases? How robust are our algorithms as they start to process large numbers of subjects? And here is just a little preview flavor of what a atlas could look like. This is using 3 subjects in 3D and looking at some tractography of the atlas around a broader area of the corpuscular symbol. So in conclusion, I've presented a novel framework for statistical analysis of structural connectomes. We represent tractography fibers as geodesics of a metric, that is, as a point on the manifold of Riemann metrics. We explicitly compute distances in geodesics between connectomes using the diffeomorphism invariant Eben metric. We provide a diffeomorphic metric registration framework to register connectomes. And we provide a structural connectome atlas building algorithm. Thank you very much for listening and paying attention to this talk. We hope you enjoyed it.